I have the great pleasure of introducing our next speaker, Dr. Kris Nichols. I was blown away by her talk at the Future Harvest Conference 2 years ago when she talked about glomalin. And from what I remember, glomalin is a glycoprotein exudate that is associated with the hyphae and spores of mycorrhizal fungi and is highly linked to and associated with soil organic matter. So I was amazed by how she was able to take something that was a very complex topic and break it down into a really interesting and still nuanced conversation. So today, Dr. Kris Nichols will be talking about soil biology and how it works. And a few things about Dr. Nichols' background. She has over 25 years of research experience studying mycorrhizal fungi. She received her Bachelor's of Science degrees in plant biology and in genetics and cell biology from the University of Minnesota, a master's degree in environmental biology from West Virginia University, and a PhD in soil science from the University of Maryland. Afterwards, she worked as a biology lab technician for 3 years, and then as a research microbiologist with the USDA ARS for 11 years. Currently, she is the founder and principal scientist of Kris Systems, which Kris stands for Knowledge for Regeneration and Innovation in Soils. Dr. Nichols is a leader in the movement to regenerate soils for the health of crops, food, people, and the planet. And without further ado, let me bring Dr. Nichols up onto the stage.
It's a pleasure to be back here on the Delmarva Peninsula. As was mentioned, I did my doctoral work at the University of Maryland. I worked for a while at the Beltsville Research Center in College Park. And I also spent some time in Pennsylvania at the Rodale Institute. So I am somewhat familiar with some of the complexities of some of the soils around here. And I currently live in Arizona, and I'm working on a project there, as well as doing some work up in Canada, and continuing to try and work on some other projects as well. And so what I'm looking at right now is this whole idea, and I really want to thank Keith for his carbonomics presentation, because it does really discuss a lot of where I think that we're at and where it is that we're going with all of these different types of systems. And a lot of things did spring out from the conversation that Keith and I had several years ago, as he mentioned, as we looked at this whole thing from an economy standpoint and how we're going to be able to look at how we can optimize soil biology to be able to regenerate soils. So what is it that we're going to utilize in looking at how carbon is flowing through the systems? I think of regenerative agriculture. There's a lot of different controversy that's going on right now on how you look at regenerative agriculture. I think of regenerative agriculture as sort of an innovative, integrated, dynamic systems. It's a whole systems approach. And it really is focusing on how carbon is flowing through the whole entire system. So it's utilizing the power of photosynthesis from the subatomic and atomic level of energy flow from the sun, as Keith was mentioning, through carbon into plants, microbes, and animals. And it's this interwoven, complex, yet elegantly simple, repeated pattern of carbon flow and how we're going to be looking at that. And when I think of regenerative agriculture from this perspective, it really is us focusing on the problem and not the issues. And so when I talk about the problem, when we're focused on regeneration agriculture, it is really about regenerating the soils. And the problem is in agriculture, I believe that there is really only one problem. Everything else is an issue. And the problem is that we do not have enough soil. And we do not have enough soil because soil is carbon, hydrogen, and oxygen, or organic matter, bound to sand, silt, and clay. And we've got sand, silt, and clay. We don't need to worry about that. But what we don't have is enough carbon, hydrogen, and oxygen, in particular the carbon, in the system to be able to have enough soil. So as we regenerate soil, as we're growing soil, this is really going to be what's going to help to energize the system and be able to move it forward. And when we're looking at this, I like to go back to thinking about how soil was actually first created, because there was no soil without plants and there was no plants without fungi. This relationship has existed for hundreds of millions of years. the relationship between mycorrhizal fungi that Keith mentioned and the photosynthetic organisms, the plants that we see today. But in an aquatic environment, it was a single-celled cyanobacterium and the fungus that created this relationship, that started this happening, that got this going to create soil. And as Keith was talking about sort of the mining that they do, these organisms are doing, that's a key component to how soil was made. Because it was that process of excuse me, I apologize, I've got a cold, so I apologize. So it was that process of making soil to begin with. And that was done by helping to get the micronutrients that the plants needed to do photosynthesis. In the beginning, there wasn't an issue with things like the macronutrients that we think about today. The main issue that was impeding photosynthetic activity was actually the micronutrients. Things like molybdenum and copper and zinc and boron. These things that would impede the photosynthetic enzyme formation and the production of photosynthetic activity. And those things impeded photosynthetic activity. So plants growing, these single-celled photosynthetic bacteria growing in this initial environment was really due to the fact that we had to have this system that was happening. Thank you, I got a cough drop. It was really important to these things that were happening in this system to get this to start to occur. And so as we were looking at trying to get this to happen, in this aquatic environment, those micronutrients were the things that weren't very readily available to the single-celled photosynthetic bacterium. So what the single-celled photosynthetic bacterium did, if you were a single cell, you just waited until you, in an aquatic environment, ended up bumping in to those micronutrients in order to be able to get them to do photosynthesis. And in that aquatic environment, there wasn't very much of them that were there. But what you did end up doing is one day you encountered a fungus. And the fungal body are these long threads. And so they have a greater, what's called a greater surface area to volume ratio, meaning that they have a greater exposure to the environment that they're in. In this original environment, it was an aquatic environment, and this greater exposure allowed them to be more efficient at getting those micronutrients because they could encounter them more. They had that greater exposure. They could reach out and grab onto them. And so the photosynthetic bacterium said, just as Keith was talking about with the carbon economy, I think of it a little bit as a trade-off analysis, but it's the same type of thing that you basically traded carbon for these micronutrients in order to be able to get those activities to occur and get that functioning that was going to be happening. Now, in this early Earth environment, as they did this and they were much more efficient, they both got to reproduce at a much greater rate. So their populations grew. And as their populations grew, they would start to wash up onto land. And as they started washing up onto land, they would all end up washing up. If you go to the shore, you see the tides only go so far, and they're always bringing things to about the same places. And so as the plant material washed up onto shore, they started to, or this pre-plant material, I should say, as it started washing up onto shore, they started to combine on top of each other. And you got this multi-layered complex that was sitting there. But if you were a photosynthetic bacterium and you were at the bottom of the pile, you had to get another job. Because you don't do photosynthesis when some stuff is piled on top of you. So the other job that happened was the formation of roots. And roots themselves are essentially anchoring structures. The idea of this is as the pile grew upwards, it became more efficient at doing photosynthesis because you had greater contact to sunlight. You were in the air, not in an aquatic environment anymore, and as you grow upwards, you're reaching towards the sun. So again, that's your initial energy source. That's that beginning. So you utilize that initial energy source, and you're being more efficient as you grow up, but as you grow up, what's going to end up happening is it's very easy for you to fall over. So the anchoring structure that the roots were created was those brace roots, or that anchoring structure, to make sure that you don't fall over. And we see that today in modern plants, where the larger you are, so very large trees, have a very intricate sort of root system, especially near the surface, that helps to brace them and anchor them in place. We'll see this also with things like corn plants or grasses, where they have those brace roots that you'll see that'll be near the surface. It's keeping them from falling over. And this was a really important process that was happening in this environment. And what's really unique about this relationship is that as this started to create soil, because the plant material was there and the fungal material that was there and the bacteria that were there, they started to build up the amount of carbon, hydrogen, and oxygen that was on land. And that started to create soil. And during this process, this relationship between the fungus and the plant stayed around. So what you see, the pictures that are in black and white, those are pictures of actually fossilized roots that are colonized by this mycorrhizal fungi. So as Keith was mentioning, sort of that black fuzzy material that's up towards the top, that's the arbuscules, these highly branched structures. The bottom picture is a high magnification of those arbuscules. And you see these highly branched structures, and that branching is very efficient. It's part of the efficiency of the design. Again, the branching allows for a greater surface area to volume ratio, which is going to allow you to have a greater exposure. So inside the root cells, you basically had a highly branched structure that would surround the cell membrane, and that would allow you to very efficiently drop off the maximum amount of nutrients that the plant would need, and to absorb the most currency, the most carbon that would come in. Again, as Keith was mentioning, carbon is your currency in this relationship. This relationship continues today, so the pictures that are in blue, stained blue there, are the roots that are other roots that are stained, modern roots that I took a couple years ago. So the whole idea here is that we're still seeing this relationship today. And although it's not necessarily playing the same role in helping all the time to create soil that it did when we didn't have soil but we just had land, it is a big part of what we're working now with trying to regenerate soil. What we're trying to do is we're trying to regenerate and grow soil from the top down and not the bottom up. So as a soil scientist, when I was going to school at the University of Maryland, a lot of the things that we would talk about with the growth of soil and the creation of soil, one of the things that's sort of soil science dogma is that it takes about 1,000 years to generate an inch of topsoil. So we can't really change organic matter, we can't really change soil in a short period of time. And this is true if you look at growing topsoil from the bottom up. The breakdown of parent material, creating that original soil, the breakdown of that mineral component, having that migrate up to the surface, and then that ending up forming organic matter and combining with carbon at that surface to create soil. And that will take a very long time. But again, what we're trying to do with this regeneration process is look at the mycorrhizal fungi, look at the growth of the plant so that we can regenerate from the top down and not the bottom up. And we can do this in a relatively short duration of time. And we're going to do this by basically doing what I think of as sort of the brown revolution. Growing soil. It's not a new green revolution that we need to have, but a new brown revolution in agriculture. Looking at that elemental issue that exists. And so what I've done is I've taken some of the soil health principles. Typically there's about 4 soil health principles that NRCS discusses. I've actually made 6. Sometimes NRCS has 5s because they include livestock. But the primary principles that NRCS has is maximizing photosynthesis, or keeping things green and growing, enhancing or energizing with diversity, reducing tillage, reducing disturbance, and armor or protection. Now again, sometimes they'll add livestock to this, but the other principle that I've added to this is reduce or no synthetic inputs. And the reason that I've added that is something that Keith was sort of alluding to and discussing a little bit as far as the jobs of the microbial community. Because if we're trying to regenerate soil, And regenerating soil is about getting carbon, hydrogen, and oxygen bound to sand, silt, and clay. We need to do that through the activities of the microbial population. And all of these principles are going to be impacting the microbial population, their diversity and their numbers, and what they're going to be able to do. And their jobs are intimately related to things that we do offset through that welfare system that is part of our introduction of inputs into the system. And we outsource the jobs of the microbial community. I really like the slide that Keith had when he was talking about this, because he talked about, low unemployment rates. The welfare system basically makes the microbial community unemployed. It's a trade-off analysis. Everything costs, nothing is free. It either is free to the plant because you spent money on putting that input in, or you're going to have the plant have to do some work in order to be able to pay the microbiology to do the job. But one way or another, the job has to get done, and somebody has to pay for it. And how can we trade so that we're not doing the thing where we're paying all of the money for the inputs? And the trade-off analysis to the plant is if the plant has to put all of this carbon into its exudates and feeding the biology, there's less carbon that is available to go into reproduction above ground or grain production above ground. And so again, it's a cost. So we have to look at the cost-benefit ratio that it is that we can look at trying to achieve. Now, I've put these principles into this pyramid design, and I did this intentionally, not just because it's kind of a cool graphic, but I did this intentionally for very good reasons. Pyramids have stood for tens of thousands of years, and they've stood because they're incredibly resilient, and the resilient architecture exists because every layer, every block that was placed in the pyramids when they were built was placed there for a very strategic reason. Everything that was done, this pyramid design is not about hierarchy. Armor is no more important than livestock. Reducing tillage is no more important than maximizing photosynthetic activity. What it is about all of these things being integrated together, all of the blocks working together and playing all of these different types of roles, and being able to reinforce the structural stability and the resilience of that system. And the other reason why I put this into this pyramid design is that we want our systems, you want your family to be able to not just celebrate century anniversaries, but millennial anniversaries. You do the most important job on the planet, as was mentioned this morning. And if you're going to do the most important job on the planet, you need to be around for as long as civilization is going to be around. So we need to be here for thousands of years. And we're only going to be able to do that if we take an integrated systems approach, looking at how we can mix all of these things together. And we're going to do this in a way that is utilizing eco-functional roles, and we're going to intensify that idea. So what took a long time, again, for soils to begin to form on our landscape, what took tens of thousands of years for this soil to be generated in the first place, we need to regenerate that soil in a few decades. And so we're going to look at the principles and the ideas of what happens from an eco-functional role within the system, but then we're going to intensify that. We're going to get that to occur at a much faster rate. And we need to do that in order to be able to maximize the efficiencies of the system, to optimize our landscape, and we're going to do that by utilizing multiple enterprises so that we don't just get income from one thing, but we get income from multiple things. That the trade-off analysis, that the economy doesn't just go from source to payment without things being exchanged on a more even manner. And so what we're going to be doing is essentially also treating the soil like we want to treat ourselves, how we're supposed to be able to be healthy. Because I said before that all of this follows some repeated pattern designs. They're intricate, but also elegantly simple, because biology follows the same types of rules. So one of the things that we want to do is that we want to eat small meals throughout the day. That's what we're supposed to do. We're supposed to be grazers. This, from a soil standpoint, is that maximizing photosynthesis. As I said, I do some work up in Canada right now, and when I'm up in Canada and I'm talking with Canadian producers, and I was recently in Colorado as well, and I go to the Dakotas where I did some work. When I'm there and I'm talking with producers, I tell them that their minimum growing season, the number of days that they have to have a plant growing on the surface of the soil, is 260 to 280 days. The sun shines 365. If that's our major source of carbon for this carbon economy, if that's the thing that's providing the energy, if you take 105 days off every year, that is not feeding the soil. That is not regenerating the soil. That's taking a really long vacation, and you should be ashamed of yourself. Here on the Delmarva Peninsula, your minimum growing season is 300 to 320 days. You need to have something growing here as much as possible. And so the cover crops that Keith mentioned, the other things that Keith was talking about, are a big part of that. It also is a big part of that to be looking at different plants and different options that you can be putting into the system. The future of agriculture is perennials and annuals and annuals and perennials and trees and shrubs. It is utilizing companion crops and polycropping and double cropping and relay cropping. It is utilizing all of these things because what we are trying to do is energize the system by maximizing how much photosynthetic activity we can do. And we need to do this because we know even in environments that are some of the coldest on the planet, there is biological activity that can occur 365 days a year. And if that is not getting a fresh carbon source, directly or indirectly from a plant that's growing, that is going to be, those organisms are going to be eating the organic matter that we're trying to regenerate our soils with. So we need to look at how much we can add, that small meals, getting things eating throughout the day, getting plants growing for as many days of the year as possible. And we also need to have a diverse diet if we're going to be healthy organisms. The diversity that's going into the soil, the different types of plants. Now, when you're trying to address having your growing season be 300 to 320 days on the Delmarva Peninsula, you're automatically going to get diversity. Because you can't just grow one plant and have that happen. You're going to have diversity that's going to be there. So again, like with that pyramid, they're interrelated and interlocked. One thing feeds into the other thing. Now, the other thing that we need to be looking at here is exercise. Exercise stress to the system is one of the most important things that we don't think about. Because we are in the jobs of growing a crop, and we want to make sure that that crop has every protection that it can possibly have. But the soil, as Keith was mentioning, has mechanisms to be able to protect the plant itself. And utilizing those mechanisms are going to be far more efficient than things that we're going to be introducing. Chemicals that we add to the system, whether they are synthetic chemicals or certified organic chemicals, anything that we're introducing to the system that is not part of how the biology is going to allow for things to flow, those things are basically offsetting and outsourcing the jobs of the microbial community. And so we need to look at how we can keep that community going. We want to exercise, but just like being a healthy person, you don't want to exercise to injury or overstress. So we want to look at the frequency of the exercise we're doing, the intensity, the scale, and the timing of that exercise. If you're going to be a world-class athlete, you don't run the same distance, the same time, all the time. You're constantly pushing yourself. You're constantly challenging the system. Part of the challenges to the system are the stresses that are introduced with things like nutrient stresses and water stresses and insect stresses and animal stresses. Grazing is a stress. Insect predation is a stress. Having an insect invasion is an overstress to injury. But you need to be managing the stressors in such a way that you're allowing for the plant to be able to work with the system. And part of the things that happen with the plant when they're working with the system with this exercise is what they're doing is the plants are creating various types of phytochemicals, of biomolecules, things like antioxidants and polyphenolics that are actually essential to the food that the animals that are going to be eating that plant material need. The way we get antioxidants and polyphenolics is from the food we're consuming. But we're losing that in the food we're consuming because we're not allowing for the formation of those molecules because we don't allow for stress. Those are stress biomolecules. Who knows what a tomato tastes like? How many of us think we know what a tomato tastes like? The majority of people on this planet have no idea what a tomato tastes like. Because a tomato, part of the flavor and texture and smell of a tomato comes from these phytochemicals that are produced in response to stress. Most of the tomatoes that are commercially harvested today are harvested green, before the plant has any way of making these phytochemicals. And we expose them to ethylene gas to get them to turn red and look like they're mature. But that is not getting the plant to make the phytochemicals that are important parts of the nutritive value of that food. So exercise and stress is one of the key components that we're missing in how it is that we take care of our plants and instead of letting nature and the system work with it. Now, the other thing is protecting our bodies, protecting the soil surface. Keith mentioned this as far as the armor or protection that we're putting on the soil surface. And that is far more valuable than anything that would happen if we were going to till that in. In organic agriculture, we do a lot of things with green manures in our crop production, and then we're going to till that green manure crop in. Why do we do that? Because we think that that's the way in which we're going to be getting the nutrients from that green manure crop. But most of what we're looking for is actually happening in the roots, and the exudates from those roots, and the biology that's stimulated and growing in response to those exudates. And the minute that we do that, we're basically going to destroy the habitats and the homes of the organisms that are in the soil. We're going to tear up fungal hyphal networks. And part of what ends up happening in this process is when we do that destruction, basically, if I'm a mycorrhizal fungus and you do tillage, you may or may not have a wrecking ball come in and destroy my home, which is the aggregate where I lived. So you may destroy my home. You may also end up ripping off some of my limbs. Now, I can live and survive limbless and homeless. I'm not going to be happy about it. And any carbon influx that I get, I'm going to have to utilize to be growing more limbs. Because my primary job, for me to get carbon, the currency, for me to get paid from the plant, I need to give the plant something. And for me to get the things, the nutrients that I give the plant, I have to have limbs. So in a tillage system, in a system where we're doing frequent tillage, we're constantly ripping off the limbs, and the mycorrhizal fungi are constantly having to reform those limbs. And when the carbon is only allocated to that reformation of the limbs, to the regrowth of the fungus, there's less carbon that can be allocated to building the home that I live in as a fungus. And the home that I live in, those soil aggregates are an important part of how things are managed from air and water and nutrient cycling. Because the aggregates are what helps to create porosity in the soil, the open space between those homes. And that open space allows for gas exchange, for air to come in, for oxygen to come in, and CO2 to escape. It allows for water to come in, and it allows for water to be held in that environment. And inside the aggregate, the reason that I created the aggregate is because it's a really efficient place for me to live in. And guess what's in there? The grocery store. I've got the neighborhood grocery store inside the aggregate with me. Because inside the aggregate, as a fungus, I help to make this home, this habitat, this aggregate, and inside there, I have organic matter and soil minerals and bacteria and other fungi. And that's all that I need to be able to release the nutrients from the organic matter and the minerals. And then I can take those up and I can convey them outside of the aggregate into the plant, and I can get carbon then from the plant and bring that back into the aggregate and utilize that and exchange that with the bacteria and the fungi to help to release more nutrients from the minerals that are inside the aggregate. So I've got my grocery store that I can keep the shelves constantly stocked, and I'll put the resources into making that, but only if I don't have to keep regrowing my limbs. So there's a trade-off. I can only have one if I don't have to do as much work here. Now, I may have to do a little bit of work here. Sometimes I may have to occasionally rebuild or regrow some limbs. And that's okay as long as I get enough carbon influx, that economy with having plants growing for a longer period of time, for me to be able to essentially then start creating the aggregates and creating that home and habitat. So again, what we're looking at is utilizing photosynthesis, utilizing the power of the sun. And as I said, we're going to do this to address the one problem that I see in agriculture, which is a carbon problem. So these are two soils. This picture comes from David Brandt, who farms near Carroll, Ohio. And essentially, the soil at the top is a soil that is how David Brandt's soil is classified. This is what his soil should look like. This is how his soil should be from his environment. This is actually the soil from his neighbor's field, which is his uncle's farm. And essentially, if you look at that, and you look at the picture next to it, what soil would you like to farm? David Brandt has changed his soil. There's a discussion as to if we should start reclassifying soil based on things that we are doing. Human impact on the soil environment. So we've got this soil that he, over 35 years, has been able to regenerate. And that is what it is that we're trying to do within this. But we don't have 35 years. We're running out of time and money very rapidly in our environment, and especially here in the Delmarva Peninsula. As I was driving over here, as I said, I currently live in Arizona, so I flew into BWI, and I was driving over here, and one of the thoughts that I had in my head was if the water goes up by 4 meters, is there going to be a Delmarva Peninsula? I don't know where it would be. I couldn't figure it out. Because we're looking at this new future. And we have the options and the opportunities to be able to change that future and change what it is that we can get. And at the same time, to regenerate soil. And when we regenerate soil, when we solve the carbon problem, not only are we going to build more resilient systems and be able to put carbon into the soil for a longer period of time and enhance biological activity, but we're going to improve water infiltration rates and water holding capacity and water quality and efficiency of use. We're going to reduce water pollution and air pollution. We're going to improve soil texture and soil tilth and reduce compaction and soil erosion. We're going to improve natural fertility and cation and anion exchange capacity and all of these things that we can do. And it is because of carbon. Carbon is an incredible atom in its ability to form all of these different types of compounds and all of these different types of roles. And if we think about this, just from an air and water quality standpoint, the best filters on the planet are carbon-based filters. If you buy a pure water system, guess what type of a filter is inside that pure water system? Is a carbon-based filter. Because the way we got clean water and clean air naturally is by the filtration through carbon, through soil. The aquifers are a natural source of clean, pure water because they filtered through the soil. If you ingest a poison or have an overdose, the first thing that they will do when they take you to the hospital is put charcoal in your stomach. Why? Because carbon absorbs stuff. It will absorb the toxins and keep them from going into your bloodstream. We utilize this technology on a continuous basis, but we don't think about it as elemental to our farm production systems. We don't think about the need to be able to have carbon in that system. So one of the things that's really changing about how we're exploring things, this is a paper called The Contentious Nature of Soil Organic Matter. I advise you all to find the paper. It's a free access paper. It's by Lehman and Kelber, and they're from Cornell University. It was published in 2015. It's called The Contentious Nature of Soil Organic Matter. I'm going to say this again because this is a paper that is changing how we view soil organic matter and what we can do to change the definition of soil organic matter. Because what we've done in the past is we've looked at soil organic matter, so we start looking at soil, and a small portion of the soil, most of it is minerals, sand, silt, and clay, a small portion of it's organic, carbon, hydrogen, and oxygen, and most of that organic component is supposed to be in this stabilized humus fraction that is actually supposed to be highly decomposed products. What we're finding is that's not necessarily the case. What we're finding is that we have a portion of that recalcitrant fraction, that highly decomposed fraction, that actually is stabilized labile organic matter. That the biology and the chemistry and the geology and the physical activities in the soil are actually stabilizing organic matter that normally in that labile fraction is going to be things like crop residues and is going to turn over in a relatively short period of time. And so this is how, as scientists, we're now getting a better understanding for the fact that when I was told that it takes 1,000 years to generate an inch of topsoil, or when I was told that you can't change organic matter percentages in your lifetime, and then I see pictures of David Brandt's farm, this is how we're doing it. Because we're utilizing the microbial fraction to actually convert the labile carbon that normally will turn over in a year or two and convert that into that stabilized fraction that now will stay around for decades or centuries. And it doesn't have to be decomposed over hundreds of years in order for that to happen. And the beauty of having the stabilized fraction is not only does this allow me to be able to build up organic matter in my soil, but it actually allows me to build up the organic matter that has the functionality that we're looking for. So in the definition of soil health, I like focusing on the continued capacity, but I also like focusing on the word function. Because that's what it is that we're looking for. We're looking for the ability to absorb water and hold on to water. We're looking for the ability to hold on to various types of nutrients, both cations and anions, and be able to hold on to them, but not make them unavailable, but make them exchangeable. And that's what organic matter is doing for us. So the functionality of that organic matter is being retained when we can keep these more labile compounds. When you're highly decomposed carbon is graphite. Graphite under pressure and time is diamonds. This is not something that is functional. Graphite is not functional. It does not absorb water. It does not hold water. It does not have a cation or anion exchange capacity. So highly decomposed material, yes, is carbon that may be stabilized for a long period of time, but it also is carbon that's not functional. How do we get more enhanced function in our system? And that's what we're doing through the biology. So it isn't just about what you get on an annual basis of this trade-off analysis of inputs versus outputs, but it's also how that contributes to the long-term cycling and availability. And when we look and think about the structure that we're forming, the aggregates that we're forming in this process and the soil porosity that we're forming, again, we talk a lot about water movement. And I think one of the best tests that you can have for healthy soils is looking at water movement, water infiltration rates and water holding capacity. And you have to put the two things together, because very sandy soil that doesn't have a lot of organic matter will have a great water infiltration rate, but absolutely no water holding capacity. Soil that has high content of clay will have a pretty poor water infiltration rate, but a really good water holding capacity. So it is that we're looking for a balance between these things. It isn't just one thing that we're trying to have that we're regenerating and regrowing in this process. And the soil aggregates are a big part of this because they change the size and the continuity, the diameter of the pores. And again, that's not just important for water movement, but it also is really important for gas movement. For the nitrogen gas to get into the soil, where the nitrogen fixers are, the rhizobium bacteria need to have the air to be able to get into the soil environment. The roots themselves, when we look at plants and we think about plants, plants give off oxygen, right? It's a really good thing. But plants also give off CO2. They respire just like we do. They have to do the same types of biochemical reactions, but they give off the CO2 from the roots. And when we don't have good porosity and good gas exchange, if your field is flooded, the plants above ground will start to turn yellow, but it's not because of the water that's at the surface. It's because of the water that's in the soil that's not allowing the CO2 to escape. So the plant is suffocating by CO2 poisoning that's happening at the roots, because the water doesn't allow the CO2 to diffuse away. So as we increase porosity, we're not just helping with water movement, but also with air movement. So it's a good thing to be able to measure. Now, one of the things that I want to talk about a little bit is this whole idea of the complexity of the consortia of all of the organisms that are in the soil. I'm very grateful to be here and to be able to speak to you, but I have to tell you that I don't understand why half the time that I get asked to come speak, because here's the deal. When I started as an undergrad, I started studying microbiology, we said we knew about 10% of the organisms in the soil. We could culture them, identify them, we knew what they did. By the time I finished graduate school, that number was about 0.1%. So essentially, I don't know why I get invited to speak to people when I went to 12 years of higher education to become 100 times stupider than the day I walked in the door. But what this tells us, what this shows, is the complexity of this environment and what's changing here. And so what this paper is focusing on, what this diagram shows, is essentially what we've got is the size of the diversity, so the size of the spheres, or the size of the diversity of the organisms that are within that group, all right? So bacteria are the most diverse, fungi are the next most diverse, and then you have various types of other groups of organisms, nematodes, mites, earthworms, and their level of diversity. Now the lines connecting these groups are essentially the interactions that are happening. Because what we found in learning about this great diversity of the organisms is not just how diverse they are, but also the fact that they're so interrelated and interconnected in what it is that they're doing. And so the size of the bars that are there, not the color, but the size, is essentially indicating the number of total interactions that happen. So between the bacteria and the fungi, there are more than 9,000 interactions where they act as consortia to provide single functions, is what this means, is one organism can't do the entire job by itself. It's reliant upon a number of other organisms. So even if we knew who all of the players were that were on the field, we would have no idea what all of the positions are that they could play, because they're all working together as a team. It's not one single function with one single organism. Now, the color of the bars connecting the different groups together, those colors, what those colors show is the percentage of the organisms within a group that are actually involved in these consortia. So between the mites, the cryptocytomatic mites, and the bacteria, that relationship is incredibly strong. Most of the organisms are involved in those consortia. So what this is showing us basically is this idea not only of the complexity of these organisms, but also how closely some of them are interrelated and how they need to have each other as part of the whole entire system. So one of the things that Keith was talking about is about the mycorrhizal fungi. And one of the things that had driven the conversation that Keith and I had had several years ago was that my spouse has a Bachelor of Science degree in economics, and so I'm a little bit nuts about fungi. I fell in love with mycorrhizal fungi when I was 19 years old, and I have yet to fall out of love with mycorrhizal fungi. And again, I'm a little nuts. And I'm going to apologize for being a little bit, no, I'm not going to apologize for being a little nuts. I'm just going to tell you I'm a little bit nuts. So I'm a little bit nuts because the only way when we got married that I got a ring on my finger was that the ring had to have some meaning. One, it's made out of recycled gold and it doesn't have a stone, because I do research and I'm out in the field and stones are annoying. And stones are things we should not be taking out of the ground. And recycled gold means that we're not taking that out of the ground either. We're using what we already have out of the ground. And on top of that, the meaning here is on my wedding ring and on my spouse's wedding ring are 2D drawings of amino acids, because proteins are made out of amino acids. And I studied a protein, glomalin, that is something that is produced, it's a glycoprotein produced by our vascular mycorrhizal fungi. And since I had to study that, we had to have amino acids on our wedding rings. Now, each amino acid has a letter. Mine spells eternal, my spouse's spells peace. And in addition, in our wedding vows, in my wedding vows, I had soil and glomalin and mycorrhizal fungi. So I'm a little bit nuts. I'm a little bit crazy about mycorrhizal fungi, and I'm a little bit crazy about mycorrhizal fungi because of the roles that they play in the system. Mycorrhizal fungi have been important to nitrogen and phosphorus cycling, but as Keith mentioned, to things like micronutrients as well. Copper, zinc, iron, boron, molybdenum, to being able to provide water, to induce the formation of antioxidants by the plants. In fact, some fungi are directly responsible for the formation of antioxidants or for the amino acids that are part of the structures of the antioxidants and phytochemicals. And so these organisms will grow inside the roots of the plants, and they will exchange nutrients for carbon. But they can also help to link various plants together. And this is one of the things that I think is one of the coolest relationships that exists. We know a lot about this relationship, but we're finding more and more of similar types of relationships between plants where you have this inter-plant-to-plant transfer. And this inter-plant-to-plant transfer is occurring because you have the linkage between the fungus growing in from one plant into another plant. The mycorrhizal fungus can grow into both plants. So the roots can't grow together, but the mycorrhizal fungus can connect them through that fungal pipeline. So we're going to talk really quick about 5 different organisms. We've got 2 plants. We've got a legume plant and a non-legume plant. We've got 2 bacteria. We've got the rhizobium bacteria that are in the roots of the legume plant that Keith had talked about. And we've got what are called phosphate solubilizing bacteria, What these bacteria are is they are organisms that are able to make unavailable phosphate that has been mineralized, that has been basically turned into phosphate that's bound to either calcium or aluminum or iron, so that it becomes readily unavailable in the soil. So they're able to release that phosphate and make it available. They produce enzymes to do that. And one of the things that they've done is, in order to make their activities more efficient, because they're single-celled and they can't move around very easily, they've attached themselves to the wall of the fungal hyphae. So that as the fungus grows, just as Keith was talking about, you have these microorganisms that will grow and move with the fungus. So as the fungus grows, it carries these bacteria with it to a new environment. Now, you have also the mycorrhizal fungus. So we've got 5 organisms, a fungus, 2 bacteria, and 2 plants, all right? So one day, the non-legume plant says to the mycorrhizal fungus, go out there into the soil and be able to get me nitrogen, phosphorus, because I have those needs in order to be able to grow. The mycorrhizal fungus says, you know what, I can do that because it just so happens that I have these phosphate solubilizing bacteria that live on my hyphae, and it just so happens that I seem to be growing into the roots of a legume plant, and that legume plant seems to have an abundance amount of nitrogen. So if you give me carbon, I will give some of that carbon to the phosphate solubilizing bacteria. The phosphate solubilizing bacteria will you give me some of the phosphate, so that phosphate can go into my hyphae, so I can transport some of that phosphate back to you. And if you give me carbon, I can then grow more into the roots of the legume plant, and I can take some of the nitrogen from that legume plant, and I can bring some of that nitrogen back to you. So the non-legume plant says to the mycorrhizal fungus, all right, here's your carbon, go ahead and go out and do the job for me. The mycorrhizal fungus says, all right, cool. So it goes over to the legume plant, and on the way it stops with the phosphate solubilizing bacteria and asks them to do more phosphate solubilization so that it can open up the hyphal wall door and it can have the phosphate flood in, and then it's able to utilize that phosphate and be able to take that phosphate over to the non-legume plant. It takes some of that phosphate on the way to the legume plant as well. Now what ends up happening is, in the legume plant, it says to the legume plant, can you give me some nitrogen and some carbon, and I will get you all the phosphate that you need to have. Legume plants are, all right, that's a pretty cool deal, I will do that. So the mycorrhizal fungus takes some of the nitrogen, takes some of that carbon, gives it to phosphate solubilizing bacteria. The phosphate solubilizing bacteria releases phosphate. Some of that phosphate and the nitrogen go into the non-legume plant. Some of that phosphate then goes over to the legume plant. Now the legume plant then goes to the rhizobium bacteria, and it says that you need to do more nitrogen fixation in order to be able to get nitrogen, not just for me, but also to give nitrogen to the mycorrhizal fungus. The mycorrhizal fungus can give some of that nitrogen to the non-legume plant. The rhizobium bacteria says to the legume that I can do that, but however, you need to have more phosphate coming into the roots, because in order for me to be able to fix nitrogen, as Keith said, it's a dinitrogen molecule. There's triple bond here, and I have to break those 3 bonds. It takes a lot of energy to break those 3 bonds. We know that very well for us up here, because up here above ground, when energy prices go up, nitrogen fertilizer prices go up, because it takes a lot of energy in order to be able to do this. For the rhizobium bacteria, what happens on an energy level is you need to have a molecule called adenosine triphosphate. There's 3 phosphate groups that are bound to an adenosine molecule, a carbon-based molecule. Now what happens is, as it goes through a cycle, one of those phosphate groups gets ripped off. When the electrons are released. Those electrons, when they pass through a membrane, are able to fix the nitrogen. It takes roughly about 16 of these cycles in order to fix 1 molecule of nitrogen. So it's a very energy intensive process, but you need to have a new phosphate group come in, because I can't take the phosphate group that got ripped off of my ATP and made ADP, adenosine diphosphate. I need to make more ATP, but in order for me to make no more ATP, I can't take the phosphate group that got ripped off. I have to have a new phosphate group that has electrons to come back in, bond to my ADP, and make it ATP. So you need to be able to get me more phosphate. Now, hold on. So now what happens is, the legume plant says to the mycorrhizal fungus, well, you've got to give me more phosphate, because I get to give phosphate to the rhizobium bacteria so that it can fix enough nitrogen so the nitrogen can go to me and nitrogen can go to you, so you can give the nitrogen to the non-legume plant. The mycorrhizal fungus says, all right, you give me more carbon, I can be able to give that to phosphate solubilizing bacteria. They're going to get more phosphate, and then I can give more phosphate to you, more phosphate to the rhizobium bacteria, fixing nitrogen, and at the same time I can give phosphate to the non-legume plant. Now nitrogen and phosphate are flowing through the fungal hyphae, but the fungal hyphae, all of a sudden there's a traffic jam, because there's too much phosphate that's in the fungal hyphae, and it can't give enough phosphate to the legume plant just for it to have enough phosphate to go to the rhizobium bacteria, as well as have enough phosphate to go to the non-legume plant. So it goes back to the non-legume plant and says, I can satisfy some of your needs, but not all of your needs. You're going to have to figure out a different way to do this. And the mycorrhizal fungus, then the non-legume plant says to the mycorrhizal fungus, here's the deal. I can give you the secret code to phosphate solubilization so that you can hold phosphate against a gradient. And then when you hold phosphate against a gradient, you can absorb as much phosphate as you need so you can give some of the phosphate to the legume plants. The legume plant can give some of that phosphate to the nitrogen, to the rhizobium, so they can fix enough nitrogen. So the nitrogen will have enough nitrogen for the legume plant, enough nitrogen to go into you. So there's enough nitrogen to then be able to go into me. And everybody's going to be satisfied. And all of this happens through a carbon biochemical language, and it happens instantaneously. And we know about this because we've done labeling studies with nitrogen and phosphorus, but we also are seeing this with a number of other different elements. And we can tap into this and utilize this to be able to utilize water far more efficiently. So not only are we going to improve the infiltration rates, this is a picture of Gabe Brown's ranch in North Dakota. This was a rainfall event in which we had 13 inches, almost 14 inches of rainfall in a 24-hour period. This is his field the next day. Now again, as I was driving around the Delmarva Peninsula, there was a lot of water standing in the fields. And I understand you've had a little bit of rain. But a little bit of rain is relative. In this environment, this is a semi-arid environment, this is basically the annual precipitation. This was the first rainfall that happened in June for the season, and it was the last rainfall until the middle of September. So again, you had to get the water in, and you had to be able to use it and keep it there. And the porosity and the aggregation are a big part of that. The nutrient exchange that I talked about is also not just nutrient efficiency, but it's water use efficiency. The plants, to have nutrients go from the soil environment into the plants, they have to be dissolved in water films. If there's not enough soil moisture, the plant will give off exudates and water to be able to manage that, because it has to get nutrients. If it is happening inside the fungal hyphae and inside the root cells and inside the root cell walls, nutrients are not lost, nor is water utilized in this relationship because it's all cellular water. The future for the Delmarva Peninsula may be that you will be underwater, but the future for much of the other parts of the United States may be that we won't have enough water. So we have to figure out how to manage too little and too much simultaneously. And the biology is going to be a big part of being able to do that.